Hello learners, welcome to my channel Biology Tutorials. I am Shrikant. Today I am going to discuss about immunological research. The today's today's modern era, one of the most promising field through to which all the research scientists, medical professionals, and health professionals looking forward with raised expectations is immunology, where they feel that there is a lot of efforts are being made to analyze some of the most important and still which is not possible to cure those diseases, and they expect that they will get a very promising and a very helpful solution through the immunological aspects of the diseases. So still lot of work has been done and is being still in progress to help the future researchers, future scientists in finding out some of the important answers to some unsolved mysteries of diseases which still remains a mystery. But with that, on one hand, it is creating a hope, a ray of hope for us that it will be helpful for modern research. Whereas on other hand, we are also facing some challenges regarding how to proceed with some of the important aspects of the modern sciences related to the immunology. So we will see here about the current challenges and its future prospects in immunological research. So let us start today's lecture. As we all know that immunology deals with the immune system of the body, and in if I say in simple language or basic language, that basically it is the powerhouse of a body's defense system, which helps to fight against some of the important pathogens and infections. Now, we all know, all the medical science students are aware that if the immune system fades or its capacity to fight against some of the important infections decreases, the diseases of pathogen overpowers the host body. Whereas if the defense mechanism of the body is strong enough, it will not allow some of the very pathogens which are very dangerous, even fatal to the host cells to spread in the body. So here, basically, the research of immunology revolves around how this mechanism works, and in this mechanism, throughout the its mechanism, what are the different steps which are taken by the immune cells, immune system, tissues, or organs related to the immune system to fight against the infections, and what are some of the important triggering responses, or how The activation of immune system takes place systematically. Moreover, whether immune system works in our body in a continuous manner or it remains or it gets activated only when there is some foreign pathogens. What are the role of some of the chemical mediators, messengers, and receptors, host receptors mechanism in the dealing with the pathogens and in inhibiting the growth of some of the cells which are harmful to the human body. So here, usually we see that it deals with prevention of infectious disease during cancer and halting autoimmune conditions. Now, autoimmune conditions is also one of the important aspects where we see that immune system get activated against its own self cells. In general, the immune system does not attack the host cells, but in autoimmune conditions, it gets activated and it damages host own tissues, organs, body. So how it happens, all those things occurs. So major problems in the field revolve around overcoming immune evasion, preventing collateral damage during treatments, and dealing with the inherent complexity of the human body. Moreover, the complexity of the human body also makes difficult to study the entire mechanism of our immune system. Now, cancer immunotherapy resistance tumor microenvironment. Tumors actively secrete immunosuppressive factors that disarm T cells. In that it does not allow T cells to function against those tumorigenic cells. And the problem is figuring out how to bypass this protective barrier without causing toxic systemic inflammation. So it is one of the most important mechanism that how they are capable of this unrecognizable, these tumorigenic cells gets unrecognizable by those cancer cells, how they bypass it without causing any toxic systemic inflammations. So if we are capable of knowing this mechanism to a great extent, we can manage the spread of tumorigenic cells cancer in the body. Next is the non-responders. Current checkpoint inhibitors and CAR T-cell therapies work well for some patients but fail for others. Researchers are trying to discover why certain cancers do not respond and how to turn cold tumors into hot ones. So here we see that some therapy, especially CAR T-cell therapies, are similarly that if you see in general also, some medicines are helpful in some patients, whereas they are not efficient in case of some different patients. So still, usually we see that hot ones are more capable or they are more susceptible to CAR T-cell therapies, whereas cold ones are not. So if we are capable to convert these cold cells to hot ones, then this therapy will equally effective against both the types of cancers. Next is the autoimmunity and immune tolerance idiopathic pathogenesis. For diseases like multiple sclerosis and type 1 diabetes, the exact trigger that causes immune system to attack healthy cell tissues remain poorly understood. Still, it is in a nascent stage that we are not able to understand that which are the factors which causes which or which which initiate that triggering response in the immune system that they start attacking their own healthy self tissues. Systemic toxicity therapies for autoimmune diseases typically involve broad immunosuppression. A major goal is finding ways to induce antigen-specific tolerance, shutting off the autoimmune response while leaving the rest of the immune system fully functioning against the pathogen. So, if such therapies start working, so we can work on both the front without disturbing the natural phenomenon of immune system, means that without causing any side effect or any negative effect on the defense host defense system, we can easily suppress the autoimmune cell. So these still the work is going on. Next important thing is the vaccine design for complex pathogen invasion and mutation. Viruses like HIV and pathogen causing malaria and tuberculosis mutate rapidly or interlatency, effectively hiding from antibodies. Designing vaccines that provide long-term sterilizing immunity against these invasive agents remains a huge hurdle. So, vaccines for HIV, malaria, tuberculosis. So, uh, if these vaccines are become available, or if these vaccines are discovered, it will be a landmark in the field of medical science because these are some of the most prominent diseases of modern world, and still a complete solution for its eradication is still not found. Though it is in today's world, they have spread very rapidly in comparison to other diseases, but preventive measures or prevention against them are very limited to them, and long-term therapy and treatment is needed for it, which is sometimes become very difficult for the patients to follow them on themselves. And for that, regular counseling and medication is needed, which is not only time consuming but also expensive. So, if for such diseases vaccine is being developed, it will be a boost to cure these deadly diseases. Next is the adjuvant development. So adjuvants are just like immune boosters that it enhances the immunity in the body without itself. We can say that itself it participates, but it boosts immune boosters. They act as immune boosters that can safely stimulate durable broad spectrum memory cells, continues to challenge vaccine logistics. Then comes the chronic inflammation and neuroimmunology, neuroimmune construct. So in neuroimmunology is also today's leading prominent field on which still the scientists are working. And in that, some of the important chronic inflammatory conditions like psoriasis, inflammatory bowel disease are linked in how the immune system communicates with sensory neurons in the gut microbiome. So, so the study, if uh, if it is decoded, that how this communication between the immune system and sensory neurons and gut microbiome takes place, it will just we will able to solve some of the very important chronic inflammatory and yeah, neurological diseases. Then comes aging and immunosenescence. So as people age, the immune system weakens and frequently generates low-grade chronic inflammation occurred as inflammation. So if any technology or any technique is evolved which can prevent immunosenescence, or we can give them supplementary immune cells, so people persons can easily during their old age can prevent with some of the low-grade chronic inflammations. Then what are the methodological and translation hurdles, preclinical limitations? Animal models like mice not perfectly mimic human immune responses. This contributes to a high failure rate when taking promising preclinical immunological discoveries into human clinical trials. Now, if the students who are aware of immunological science or any other medical sciences, we see that in such cases, mice is the one of the common experimental animal in which such type of immunological tests are being performed. But the issue is that they do not exactly represent the immune system of the humans. So sometimes any of the therapy or any of the treatment we study or any clinical trials which we study on any animal model, so we are not able to get the exact result about that. So that is also a very challenge in front of today's modern researchers, that how we can overcome this issue so that we can get a very precise and accurate result of whatever the experiment is going to be performed. Next is clinical heterogeneity. So no two human immune systems are identical, causing high variability in how individuals respond to therapies, making biomarker identification. But obviously, we know that each and every individual is different, so it becomes very difficult to understand that which type of treatment is suitable for one person and which is unsuitable for other. So these are some of the important immunological research which was measured by this, including the high failure rate of translating preclinical findings to human trials, the extreme inter-individual variability of immune responses, and immune redundancy, where backup pathways compensate for targeted therapies. The field also struggles to manage severe adverse events like autoimmune immunology and lack of universal biomarkers. These complex challenges require multi-disciplinary approach across several key areas. First is the translational bottlenecks and preclinical models. The primary bottleneck in translating immunological breakthroughs such as checkpoint inhibitors into clinical reality is the inadequacy of current preclinical models. Animal models often fail to accurately replicate the complex spatio-temporal dynamics, multi-organ interconnectedness, and genetic diversity of the human immune system. This leads to translational failures when moving from animal testing to human clinical trials. Next is human heterogeneity and redundancy. As we have already discussed, that each and every individual has its own unique genetic and functional variability. And this inter-individual clinical heterogeneity causes varied responses to vaccines, immunotherapies, and infectious diseases. Furthermore, immune redundancy, where multiple overlapping molecule pathways perform similar functions, makes it incredibly difficult to design highly specific therapies. Targeting a single cytokine receptors may, may prove ineffective if a redundant pair compensates for the loss. Next is autoimmunity and related toxicities in the field of immunology. A major research challenge involves harnessing the immune system to kill cancer cells without triggering severe autoimmune reactions or adverse immune events. When researchers use therapies like checkpoint inhibitors to strip away the immune system's brakes to attack tumors, there is causing the immune system to attack healthy tissues instead. Finding the balance between effective tumor eradication and the prevention of autoimmune like toxicity remains very useful. So both the functions quite difficult that on one front, if you want to destroy tumorigenic cells, there are chances, obvious chances are there that it will also destroy healthy cells. So it remains a big challenge to find a solution or any technique, method through which only tumorigenic cells destroy, whereas the healthy cells remain intact. Then comes methodological complexity and data integration. The immune system operates across multiple levels, from biomolecules and single cells to organs and the whole organisms. Traditional reductionist approaches often fail to capture this systemic complexity, while researchers utilize vast amounts of single-cell sequencing and spatial biology data to identify predictive biomarkers. The field struggles with standardizing experimental pipelines, integrating multi-modality data, and minimizing computational errors. So this is not related completely to the experimental part or the immunological cells in functioning, but it is something which is related to the, we can say, the advancing the modern technology to study the functions or their approach towards the uh, host cells or the foreign pathogens. So still there is difficulty in finding the some of the advanced technologies or instruments through which we can understand the exact mechanism of its functioning, as well as the proper utilization of its data and to avoid the error part. So this is also one of the important aspect. Then complex diseases and pathogen evasion, developing effective therapies and vaccines for rapidly mutating viruses, as we already discussed, or tackling chronic inflammatory diseases remains a daunting hurdle. Pathogens have evolved sophisticated mechanisms to evade or suppress host immunity, while chronic conditions like autoimmune diseases and allergies still lack complete understanding of their exact pathogenic mechanisms. Researchers and data scientists are continually addressing these bottlenecks by advancing computational immunology and utilizing sophisticated artificial intelligence to analyze and create high-dimensional immune data. So, still work is under the process to overcome this technological glitch associated with immunological study. And with the advent of artificial intelligence, we are hopeful that all these hurdles, all these difficulties will be overcome in coming days. Next important thing is that immunology faces major hurdles like translational failures. I wanted to discuss that. Mouse models not predicting immune outcomes, clinical heterogeneity, immune system redundancy, Then researchers remedy these issues using advanced computational modeling, human-derived 3D tumor immune organoids. Now, this is one of the important aspects that this immune organoids is one of the is considered is one of the future prospects in studying the immunological field, and this is the 3D, three-dimensional tumor immune organoids and single cell omics, ultimately accelerating precision medicine and targeted therapies, which will obviously increase the precision and the target-based therapies. The most effective cutting-edge remedies deployed to overcome current immunological research barriers include patient-derived human organoids. The challenge is that traditional 2D cultures and animal models often fail to replicate the complex human-specific tumor microenvironment. The remedy is the scientists now integrate immune and stromal cells into 3D patient-derived organoids. These serve as preclinical avatars that reliably stimulate the human immune system, allowing researchers to evaluate immunotherapy efficacy and predict patient-specific responses before human trials. Artificial intelligence and multimodal data integration. The challenge finding the right patient cohorts for clinical trials is costly, and the high dimensionality of biological data causes translational roadblocks. The remedy is that the pharma is heavily utilizing AI-driven tools such as structural prediction models and in silico perturbation to identify novel drug targets, anticipate adverse immune toxicities, and repurpose approved drugs. This helps streamline protocols and decode the complex interactions among tumor, immune, and stromal cells. Single-cell and spatial transcriptomics. The challenge is the immune system's high level of individual uniqueness and redundancy causes generalized treatments to fail in various patients. The remedy is that researchers use single cell and spatial profiling to pinpoint exact subsets of immune cells at the tissue level, allowing for targeted therapies and personalized reprogramming of innate cells. Solving immuno-oncology challenges requires tackling the immunosuppressive tumor microenvironment, therapeutic resistance, and tumor heterogeneity through precision medicine. Key solutions include using multi-specific antibodies, exploring neoadjuvant window of opportunity studies, employing AI predictive neoantigens for personalized mRNA vaccine, designing allogeneic off-the-shelf cell therapies. Current hurdles and their specific actionable solutions include overcoming the immunosuppressive tumor microenvironment. The challenge that tumors actively evade immune detection by infecting regulatory T cells and myeloid-derived suppressor cells, rendering checkpoint inhibitors ineffective. The solution for this is to adapt adoptive cell transfers to enhance cell tumor interactions or utilize combination therapies that include oncolytic viruses and targeted kinase inhibitors to actively break down the immunosuppressive stroma. Targeting cold or heterogeneous tumors. The challenge is many solid tumors lack sufficient T cell infiltration, making them cold, or contain diverse cell mutations that allow them to bypass treatments. Improving patient stratification and biomarkers. The challenge efficacy remains unpredictable, and standard biomarkers like EDL1 expression or tumor mutation burden fail to accurately predict outcomes for a majority of patients. The solution is to implement longitudinal individual patient follow-ups and utilize biospecimen models like organoids to predict human therapeutic targets prior to extensive clinical testing. Addressing severe toxicities, the challenge with therapies like CAR T cells can cause cytokine release syndrome, while checkpoint inhibitors can cause severe immune-related adverse events. The solution is that proactively manage toxicities using biomarker-guided drug holiday protocols alongside co-administration of target anti-cytokine therapies like IL-6 inhibitors for CRS. So here you can see through this diagram that how organoids or three-dimensional cell structures are derived from stem cells. So this is the current scenario of the immunological research, and there are several issues which are needed to be addressed to come to the conclusion. But still, the scientists are optimistic that the research will proceed in a positive direction. And also, as we have discussed, that there are multiple types of complexities which are associated with finding the important aspects of diseases or their solutions. But still, the modern technology, uh, the new innovations, and the, the, the some of the important findings on immune functioning of immune cells will certainly help the scientists here to come to some certain conclusions and help us to treat some of the challenging diseases, some of the important diseases which are still remains a challenge despite they are still remains for a long term in our world. So despite all those hurdles and bottlenecks, the immunological research is a promising field that the solution or treatment for all such diseases will be found out. So with this, I conclude my lecture, and I hope that to all of you, it would have helped you to gain knowledge and to understand the future prospects in this field. If you like my lecture, kindly like and subscribe my channel, which will keep me motivated to bring new videos with current scenarios of modern sciences, or if precise, I can say multiple sciences. Thank you.